Okay, so I'm coming from Bordeaux, and some of the results I will show you have been obtained in Bordeaux. And now I work in Lyon, in the Institut Lumière-Matière, the Institute for Light and Matter. And I will show you mainly examples of what we can do with whatever you call it, laser ultrasonics, optoacoustics, and many of the things that have been presented this morning. I will show you some examples on single cells, how we can use that to image the mechanical properties of single cells. The aim here is not to obtain, like in photoacoustics, images based on the optical contrast of the tissues, but images with mechanical properties as the optical as the contrast. And the purpose for this is that cells, as tissues, are mechanical objects that are their mechanical properties dictate their behavior, and their behavior influence their mechanical properties. There is a feedback all the time. So to simplify, a cell is composed mainly of two components. There is the nucleus that you can see in blue on this image, and that's the brain of the cell. That's where all cell activity is directed. That's where DNA replication and gene activity is regulated. And this is surrounded by a skeleton that you can see in green. You have fibers that act as the hands and feet of the cell. That's how it moves. That's how it's changing shape. That's how it's dividing. And these two elements are mainly composed of fibers, biopolymer fibers. It's obvious on the cytoskeleton. And these fibers are entangled, and they form a highly cross-linked network that can, for instance, in the nucleus, open or close to regulate gene activity, or in the case of the cytoskeleton, that can assemble or disassemble in order to change the shape of the cell and allow it to move. And these two components are linked together by mechanical and chemical links that allow them to communicate through these two channels. And the cytoskeleton is also linked to the environment, that is either the extracellular matrix or an implant that you want to put inside the body, a bone implant for instance, and also with mechanical and chemical links. And the cell only attaches to the environment through specific locations that through which forces can be transmitted. So you have the cell on this part and the environment. It only attaches through this adhesive patch, and forces can be exerted by the cell through these mechanical links, but also the environment can apply forces to the cell. And these forces are kind of sensed by the cell, and then they are transmitted to the brain of the cell, to the nucleus, through this mechanical chain of communication. And then the nucleus can take the proper actions given the information about the elasticity or the mechanical properties of the environment. One consequence of this is that if you have any alteration of the mechanical properties of the cell, then you will have a dysfunctional behavior of the cell. An example of this is lamellopathies, which correspond to an alteration of the proteins that form the membrane of the nucleus. And when you have that, you have a perturbation of the mechanical structure of the membrane of the nucleus, which doesn't allow for the cell to remain in a controlled shape and in a controlled mechanical state. So if you take a healthy cell with a healthy nucleus, in the left column, and if you apply a stress to this cell with the laminar flow, for instance, without the flow or with the flow, the cell is able to resist the strain and maintain its shape. But if you take a lamin deficient cell with a membrane that is not able to resist the stress, when you apply the flow, with no flow the nucleus is relatively round, but when you apply the flow, it starts to elongate and have a more elliptical shape, which means the network of fibers which is inside the nucleus has to deform to follow the shape of the nucleus. That means the gene activity will be changed because the nucleus is changing shape. And that leads to many problems like muscular dystrophies and many diseases. So a change in the mechanical properties of the cell leads to disease. This is also true for cancer. The specificity about cancer cells is that they are able to invade healthy tissues. They are able to deform highly and go through small spaces like a rat and invade healthy tissues. And this is because they are softer or more deformable than healthy cells. If you look at the Young's modulus of cancer cells, it is smaller than healthy cells. And this is this property, this abnormal property, that allows them to be so invasive. So that's for the mechanical properties, but also, as I said in the beginning, cells are able to sense their environment, the mechanical properties of their environment, and this is obviously very important to optimize cell adhesion. A famous example of this is that when you take a substrate with different rigidities, a gradient of rigidities, you have on this substrate, you have the upper part, which is relatively soft, 4 kilopascals, Young's modulus, and another region with 70 kilopascal Young's modulus, so more rigid. And you randomly place cells on this surface, and you allow them to migrate freely. And you wait a little bit, and all the cells will move to the most rigid part of the substrate. They're able to sense the rigidity of the substrate, and they're able to choose which part of the substrate is the best for their development. So the cell is placed on the substrate and is constantly sensing through these adhesive patch, sensing the mechanical properties of the environment, sending the information to the nucleus, and the nucleus says, no, this is too soft. Try to find a better location. So the cell moves until it finds the right spot, and then can continue its life there. And another consequence of this is that, for instance, if you want to place stem cells inside the body in order to do tissue replacement, and you hope that these stem cells will differentiate into the right cell type. If you want to replace brain, you hope that the stem cells will differentiate into neurons, for instance. Then you need to put inside, before you implant the cells, you need to put a matrix that has the mechanical properties of the tissue you want to replace. So you put a matrix that has the mechanical properties of the brain, then you inject the stem cells. They will recognize that the environment has the rigidity of the brain, and they will naturally differentiate into neurons. So these quantities are very important to measure in order to first understand how it works, and secondly, potentially to be able to control these properties, either to optimize cell adhesion for the development of implants, or to be able to change, to tune the mechanical properties of cell to cure diseases. So, to this aim, many techniques have been developed in the past, and mostly inherited from material science. The most commonly used probably is AFM, with which you would tap onto the surface of the cell with a tip, with a sharp tip, a small tip, and you press onto the surface of the cell, and you record how much force you need to apply to indent the cell out that much. And then you can extract the Young's modulus. You can do the same thing with pipettes, with which you apply a flow of liquid, and you push the cell inward, and you measure how much deformation you can get with a given pressure. You can also suck the cell, do the contrary, and look at the deformation inside the pipette. You can also place the cells in between two parallel plates, stretch them or compress them, like in a classical tensile stress, and see stress test and see how much force you need to apply to deform the cell. But all these techniques require direct contact to the cell, and the problem with that is that you don't know if the mechanical property you extract is an intrinsic property of the cell, or if it is a reaction of the cell to what it considers to be an aggression from the outside. Like when you apply the flow to the nucleus, it resists the deformation just to maintain its shape. And these measurements are difficult to make because also the structure of the cell implies that the dynamic behavior is very complicated. If you look at the shear modulus measured either in the nucleus or in the cytoskeleton as a function of frequency, you get these kind of curves that resemble that you would obtain in polymer Because you have these fibers inside the cell that are entangled, you have a very complex rheological behavior. And one way to measure this is to, for instance, insert beads, small beads inside the cell, and look at the Brownian motion of the bead. If you follow the Brownian motion, and using Stokes' law, you're able to compute, to extract the shear modulus as a function of frequency. And you get these curves in the nucleus, in red, or in cytoplasm. And if you look at the curves in the nucleus, for instance, at low frequencies, you have the elastic modulus, the elastic part of the modulus, and the viscous part of the modulus, that corresponds to dissipation. And at low frequency, the viscous modulus is much larger than the elastic modulus, so we're in a regime where the cell is like a liquid, as we saw in the previous days. But if you increase the frequency, the elastic modulus starts to dominate, and we are in a regime where the cell is like Homer Simpson, something in between a liquid and a solid. And if you increase the frequency even further, you reach a glass transition, beyond which the cell is an elastic solid. Not a crystalline solid, an amorphous solid, pretty much like a soft glass. But in this regime, you can have compressional wave and potentially shear waves, because the cell is elastic. But you have to go to frequency high enough, and if you look at the scale, it's not so high, only a few hertz, and you're in this regime. Regarding adhesion, you can use mostly the same techniques. AFM, you can push the cell and see how much force you need to apply to detach it. You can also use microstructured surfaces, like in this case you have an array of pillars, soft pillars that are able to bend. You know the bending rigidity of each pillar. And when you place the cells on top of the surface, it will adhere to the adhesive patches and it will bend the pillars. And if you record the bending of the pillars, you know the bending rigidity, you can measure the force exerted by the cell. You can also do a similar thing with traction force microscopy here. You have a substrate, soft deformable substrate, an agarose gel or whatever hydrogel you want. You put fluorescent beads in it and you place the cell on top. And when the cell adheres, it deforms the substrate and the fluorescent beads move. And then you track the position of the beads and you're able to reconstruct the stress field inside the substrate. That's what you see with the arrow. You can see here there's a cell here, the beads have moved compared to this position. The disadvantage of these two approaches is that you need to engineer a special surface to be able to measure the adhesion, which is not always compatible with what you really want to do. If you want to study the adhesion of a cell onto a metallic bone implant, for instance, obviously you cannot use this technique. So, because we're talking about ultrasound, we can think of it as an alternative technique to perform such measurement in a non-contact way, because it will be non-contact. We'll also have a contrast based on mechanical properties. That's what we want to measure here. It will be non-invasive, non-ionizing. Potentially it's cheap and fast. In the present case it's not, but it could be. And we also need to take care about the resolution we can have, because a cell is quite small, and the classical image we get using ultrasound is this one, where you have tens of megahertz frequencies with a resolution of a fraction of a millimeter, which is not small enough. You can use the photoacoustic techniques, but you will get a contrast which is based on the optical properties, and you will still not get quite the right resolution. You will be limited to tens of microns. So you need to go a little bit further in frequency. So you could use acoustic microscopy, in which you focus the sound with a specially engineered lens, and you can reach several gigahertz frequencies at room temperature and obtain such images. But you can see it's still quite not enough, because if you compare the optical image to the acoustical one, it's a little bit blurry. And the structures you want to image in the cells are much smaller. For instance, the fibers that are composing the cytoskeleton are only 30 nanometers in diameter. That's what you see in green on the fluorescent image. And the adhesive patches that you find mostly on the edges of the cell in this image are 10 to 100 nanometer. So you need higher frequencies. And one way to do that is to use the same principle as in photoacoustics or as in laser ultrasonics as Chris just presented, is to use a laser pulse to generate an acoustic wave, but using much shorter pulses, femtosecond pulses, that will generate an impulse-like response of the material. So you send a short femtosecond pulse to the sample, it will absorb it, it will heat up very fast, and generate an acoustic wave, but on a much smaller, shorter time scale. And the main difference with what we have seen previously here is that the pulse duration here doesn't matter at all. It is so short that you can consider it to be a Dirac function. If you look at the acoustic, well, first, you send the light, you focus it into a metallic or something that absorbs light a lot, and it will be absorbed over a few nanometers, say 10 nanometer in a metal. So the acoustic, the optical depth profile will look something like that. An exponential decay inside the depth of the material with characteristic length of 10 nanometer. And the acoustic wavelength you will generate will be of the same order. If I have an optical skin depth of 10 nanometer, I will have an acoustic pulse with a center wavelength of 10 nanometer. If I convert this into time just by putting in the sound velocity inside the metal, I get typical frequencies of 500 gigahertz. And if I'm able to tune the absorption depth of light, I can go even to terahertz frequencies. So if I compute the wave equation with all these parameters and I plot the acoustic strain as a function of time, I get a pulse here that is only 4 picosecond wide. This is much shorter than what we have seen before, but it's also much longer than the pulse, the laser pulse duration. That's why we don't, this is not important. The pulse duration, the laser pulse duration has no influence here. The acoustic pulse we generate is so short that only the optical skin depth matters. That's the main difference. So this we have seen. We have high acoustic frequencies, so we need to detect them. And the most efficient way to do this is to do it optically. This is the classical way. You have your laser beam that you split into two beams. This is the pump beam that generates the acoustic waves, and the other beam is the probe beam that you can delay in order to probe the thermoelastic response of the sample at different times. You have the pump beam, pump pulse that arrives here, the thermoelastic response of the sample, and the probe pulse that you delay by moving this set of mirrors, and you probe the response of the sample point by point. Each position of the mirror corresponds to one sampling time, and by moving discretely the mirror, you're reconstructing the whole response of the sample. You get a very high time resolution, obviously, the resolution of the laser pulse, but it's very time consuming. Typically, if I'm honest, to get a complete signal at one point with this technique, you need to scan over several meters the mirrors, and it takes about 1 hour just to get one point with enough signal to noise ratio. The way the probe interferes or interacts with the sample is what we have seen this morning. It could be interferometric techniques or photoelastic interaction. I will come back to that later. But to give you an example, this is the kind of signals we usually get. At 0 time delay, when the pump and the probe pulses arrive at the same time, you get the sharp rise, followed by a sharp decay, and then a slow tail that corresponds to microscopic heat diffusion. And this sharp rise is also specific to using short laser pulses. It corresponds to the ultra-fast the electrons that are excited by the photon initially and that diffuse inside the material. The first consequence of this is that the electrons will diffuse and broaden the initial size of the source, so you will get a broader source. And also you will have free negative carriers. So if you place a liquid or cell on this surface and you have electrons, you have a risk of doing the sort of a hydrolysis of the cell and generating hydrogen, which the cell would interpret as a trigger to commit suicide, which is called, in a more fancy way, apoptosis. It's a programmed death process inside the cell, which will be triggered by these negative carriers. So you want eventually to avoid this. So all in all, this technique potentially has a good lateral resolution, 1 micron, which is compatible, which is limited by the ability to focus the beams with the lens. We have a typically 100 nanometer in-depth resolution due to the high frequencies we can generate, which are limited by this electronic diffusion that broaden the source. And if you're able to engineer the proper transducer, it is non-contact. So I will try to answer just two questions actually, which are: what can we learn about the structure of the cell from the propagation of acoustic waves at such high frequencies and small length scale inside the cell? What can we learn about the structure of the cell? And how can we perform single cell imaging with this technique? So before I go to imaging and before I go to animal cells, I will start by plant cells, because they are simpler, because they don't have the cytoskeleton that we saw on the green, in green on the fluorescent image. They are much cheaper. We can buy it at the supermarket. And they're easy to work with because you can work at room temperature for a couple of hours because they are dry. Of course, you will have a certain smell to it in the lab, but it's cheap and you can have it easily. And they're composed of mainly the vacuole, which is a balloon filled with pressurized water. It is pressurized, the turbo pressure, to allow exchange with the environment. And all around this vacuole you have the cell wall. There's a rigid structure, anisotropic arrangement of cellulose fibers that is here to contain the pressure and to allow the cell growth, but only in given directions. And if I look at the top view of onion cells, a strip of onion cells, you can see they're quite elongated. Maybe it's no. Okay, they're elongated because the cell wall is anisotropic to allow cell growth in only one direction, and that's why the plants mostly grow upwards, because the anisotropy of the cell wall.
What is the size of the cell?
This is hundreds, or tens, several tens of micrometer in this direction, hundreds in this direction, and the thickness of the wall in the thickest part here is a couple of microns, 1 or 2 microns. And the size of the cellulose fibrils is tens of nanometers, so much smaller scale. And if you try to measure the mechanical properties of this structure with the contact techniques I showed you previously, for instance FM, if you try to press on the surface of an anisotropic layer resting on the pressurized liquid at just one single point, and if you don't know the mechanical symmetry of this anisotropic structure, there's not much information that you can extract. So that's where acoustic wave can be useful. And to work with this sample, we just designed a very simple basic transducer that is made of a thin metallic layer, typically 300 nanometer thick, made of titanium because it is biocompatible. And we spotted this layer on a transparent window that can be glass or even better sapphire. And we focused the pump beam that generates the acoustic wave at the bottom of the metallic film to avoid the free electrons I was talking about. If you will generate electrons, it will be in this region, and the cell are on the other side, far away. They are shielded by the metallic film from the free electrons. And the heat generated by the laser will of course diffuse inside the metal, but if you have as a backing a highly diffusive material such as sapphire, the heat will mainly sink into the transparent backing. So we also prevent overheating of the cell due to the laser. The other thing you can do is that, as we have seen before, what makes the sound generation is the ability of the material to dilate after the temperature rise. So if you choose carefully the ratio of the rigidities of the metal film and the transparent backing, you can also optimize the thermal dilatation and improve acoustic generation. So what happens in this case is that we have the pump pulses, which are focused at the bottom of the metallic layer. It's up, dilates, it generates an acoustic wave that propagates through the metal film and reaches the cell where it is partially transmitted. So you have an acoustic wave that propagates inside the cell. Then you use the second part of the beam, the probe beam, that you focus through the cell, and it is reflected at the cell metal interface. So you have a first beam that is reflected, static beam, if you wish, and there is another beam that is backscattered by the acoustic wave that propagates inside the cell. So you have two optical contributions that we send to a photodiode, and as the acoustic wave moves, you will see an interference pattern that also moves depending on the position of the acoustic wave. And the faster the acoustic wave moves, the faster the interference pattern will oscillate on the photodiode. So that's a direct way, provided you know the optical index of the cell, that's a direct way to measure the sound velocity inside the cell. So we do this on these onion cells. In two configuration, we either place the cells with the vacuole facing downwards in contact with the transducer, so in this case we are probing the vacuole, or we flip it over and place the cell wall in contact with the transducer. And in these two configurations we get oscillations because of this interference pattern. In the case of the vacuole we get an oscillation at 6 GHz, and in the cell wall we get 12 GHz. A higher frequency means a higher sound velocity, so a higher rigidity. So just to be more quantitative, we do that on several cells. We build distributions of values, and in the vacuole we get a sound velocity of about 1600 meters per second, which is close to that of water, as expected. And for the cell wall, we get a sound velocity which is higher, about 3400 meters per second, which we can convert into rigidity, which is about 13 gigapascal, very high. And this is so high, it's almost that of pure cellulose. And this is surprising because the cell wall is composed of cellulose, but not only, there's also a pectin matrix that embeds the fibers. So from a mechanical point of view, you would expect the two components to influence the average rigidity you measure. And given the structure of the cell wall, you have the fibers, which are roughly aligned in this direction, and you are compressing with the acoustic wave in the other direction. So you can model this as a stack of layers of cellulose, pectin, compressed in this direction, an isostress material. And if you calculate the effective rigidity of such material, you find a rigidity of about 7 gigapascals, given the values of each layer. This is much smaller than what we actually measure because we are missing an ingredient here. It is the fact that the fibers, which are in different planes, are connected together by xyloglucan bonds, and they form a high cross-link network that spans over the whole thickness of the wall. And that's why what we measure is only the influence of this highly cross-link network, and we don't see, or barely see, the influence of the surrounding pectin matrix. So this goes to show that such measurement can give information about the structure of the fibrous network that is inside the cell. So if we're able to apply this to the nucleus of an animal cell, for instance, we're able to sense the conformation of this fibrous network inside the nucleus. And when there is a change due to the change of gene activity, we can follow this just by measuring the sound velocity inside the nucleus. So the next step is to do images. So in this work, it's the same transducer, titanium sapphire for heat diffusion, the cells on top, pump, probe, and then you just scan the lasers across, perform an image. At each point, you get this oscillation due to this interference pattern, and you plot the amplitude of the oscillation at a given time. And you do this for all the pixels, and you get an image. And that's what they show here. So you have 3 images at different times, because each time corresponds to a specific location of the acoustic wave inside the cell. So if I look at the amplitude of the oscillation at, say, 200 picosecond after the excitation, this image, I'm somewhere 300 nanometers away from the metallic surface. And as the time goes, 300, 400 picosecond, it's a little bit further in the cell. So you get these images, which are nice, but you think, maybe I can improve the contrast, because it's a little bit blurry. So one way to do that is to work on the transducer and try to engineer something a little bit clever, more clever. And so in this case, it's upside down. The cell's at the bottom, and pump and probe beams are both coming from the top. And you have this structure that we also have seen in Paul's presentation, which is made of a dielectric layer sandwiched in between 2 gold layers. And the purpose here is to maximize pump absorption, to limit pump transmission in order to maximize the generation, but also to shield the cells from the laser radiation. And you also want the same transducer to transmit as much probe as possible to maximize the detection. So what they did here is to compute the transmission of such structure as a function of the gold and thickness and ITO thickness, and that's the pump transmission, probe transmission. So regarding the pump transmission, you want to be in this region where it is as small as possible, whereas for the probe you want to be here, where the transmission is the highest, until they find a compromise between the two at this location. And the other thing you need to take care of is the mechanical resonance of the structure. This is a simple film, and it will resonate at the natural frequency, and in this case it is about 10 gigahertz. And this frequency must be different from the oscillation frequency you want to measure, otherwise it will overlap, and you won't be able to tell which is which. What's this transducer? Perform the same images as before, scan the lasers across, and this is the image they get, which is nicer. You can see the small links in between. This is one cell, this is another one here. You can see the links here between the cells that you find on the acoustic image. You can see contrast inside the cell. You can see a region which is more rigid, well, which has a higher sound velocity, and that corresponds to the cell nucleus that you can find here. So that's the first way to do it. You can do sectioning inside the cell just by changing the time, but you can also do it in the frequency domain, but using a confocal microscope. So this is more a classical Brouillon spectroscopy, for those who know it. Instead of doing it in the time domain with this data line, you do it in the frequency domain directly using a spectrometer. So you shine a laser on the sample, it diffracts the light, the sound inside the sample diffracts the light, you record it with the spectrometer, and you're able to see at which position, at which frequency the interference, the maximum. And they send it into a confocal microscope and do confocal sectioning. And they obtain such images here, where they have different planes at spacing of about 1.5 microns. This is the first drawback, is that the confocal sectioning is much less, you have a lesser resolution than in the time domain. In the time domain you can do sectioning every 100 femtosecond. And this is close to the interface, so you can really see the shape the outside of the shape, and as they move up inside the cell with this confocal sectioning, you see less and less, and you finally only see a round area, which is the thickest part of the cell that corresponds to the region where you have the nucleus. So from the side you have a bump where the nucleus is. And they did a very interesting test. They introduced a drug to inside the cell, the cytochalasin D, which has the property to depolymerize the actin, the cytoskeleton of the cell. So initially you have a cell which is very cross-linked network of fibers inside the skeleton, and you apply the drug and it will disassemble and fluidize. And as the cell fluidizes, the sound velocity decreases, and they see it as a shift in the frequency of these interference. So they're able to sense here the variation in the conformation of the fibrous network inside the cell. So that's for the mechanical properties. You also want to be able to measure the cell adhesion. So just to do that, you change a little bit the configuration, not so much. You just flip the probe that was coming from the top through the cell first. You flip it to the bottom also with the pump beam, and you have the two beams at the bottom of the metallic plate. This is even better because the cell doesn't see any laser radiation. And the signal you get is the one I showed you previously. You have sharp rise and acoustic echoes that bounce back and forth inside the metallic plate. So the first echo you see here corresponds to the acoustic wave packet that has been generated at the bottom here, that has propagated through the metal film, reflected off the interface, gone back to the bottom, and it is detected there. So the amplitude of this first echo contains the information about the acoustic reflection coefficient at this interface. So potentially it contains information about adhesion. And if I look at the amplitude of this echo at different location, below the cell or outside, I see that I have an amplitude which is larger when I'm outside the cell because less acoustic energy is transmitted to the cell. So if I just monitor the amplitude via this acoustic echo at functional position and plot this amplitude, I will get a map of the acoustic reflection coefficient. So this is what we do here. So, okay, just to mention that in this experiment we're using, we're not using the delay line thing, we're using the asynchronous sampling that has been shown this morning, but I just would like to add something. It's that, okay, so you have these two trains of pulses that have slightly different frequencies. So you have the pump pulses here. Each pulse generates a thermoelastic response in the sample, and the probe pulses are initially synchronized with the pump pulses, and they're increasingly being shifted, and at each probe pulse probes the thermoelastic response at a later time as they are shifted to this asynchronous sampling. And in this case, we just have a slight shift of 500 Hertz in between the two train pulses, which means we are able to reconstruct the whole response in 1/500 Hertz. In 2 milliseconds, we have the whole sample response in comparison to 1 hour with the delay line. So this is a huge reduction in the acquisition time that allows doing images much faster. If you want to do an image with a delay line with a 1 hour per point, it's going to take forever. This is still very long, but it is much faster. So this is what we do, and we do it under this cell, the same cell that you've seen in fluorescent microscopy, and I'll just show you this image. So we image under this part of the cell where you have the fibers inside the skeleton, and you have the adhesive patches at the edge. And I just record the amplitude of the first echo, and I play it in time for each pixel, and I get this moving. Maybe if it works. So when the first acoustic pulse arrives, you will see the contour of the cell there. You start to see the fibrillar details, and the contrast decreases because the acoustic pulse is moving away from the interface where it is detected. Then the contrast will go back as the second acoustic pulse comes here. It is reversed because the acoustic pulse has changed sign in between two reflections. So, try to pose it at some.
How do you, what kind of electronics do you have with a resolution of 1 picosecond of the process? How do you process the, in terms of electronics, to get time of 1 picosecond?
In this case, it is the.
You understand my question?
Yep. When you do this, your so pump pulse, the response of the sample, which is very fast, and you have this picosecond time scale. But when you shift, when you shift the probe pulses, what you get is a dilated time scale. You don't reconstruct that, you reconstruct this one, which is much slower, and you're able to probe it.
The detector is a photodiode, correct?
Yeah, but you can use a photodiode in the megahertz range.
In a megahertz range?
Yeah, because you're just you have 1 point every repetition, every pulse, and the repetition rate is the repetition rate of the laser. Typically, it's 50 megahertz. So you need to have a photodiode which is faster than 50 megahertz. And if you use the data line, you're just sampling at a given time. You stop the mirrors, so you're just probing at one time, and you can integrate as long as you want. So, okay. So I just wanted to show you the details of the movie, if I can. Keep moving the mouse, otherwise it disappears. Oui.
I have to point to the pause button. Yeah, okay.
Yeah, but if I don't move the mouse, it disappears in control. Well, this is not the best time, but you can see a little bit of the fibers that you see here on the phase image. Wow, too bad. So you can see the fibrillar details inside the cell. You can even see the beginning of the cortex where all the fibers are entangled around the nucleus. And I don't know how much time I have left.
Yeah. We have now 42 minutes.
Okay. So quickly, we have an acoustic pulse, so we can analyze the frequency dependence of the acoustic amplitude and transform just with a simple time frequency analysis. We can convert this time pulse into an acoustic reflection coefficient as a function of frequency. And if we look at that, at different points inside the cell, we see it depends on the frequency, which is unusual because if you send an acoustic pulse across an interface, a perfect interface, it will be reflected with a constant amplitude throughout the whole spectrum. So if you have a frequency dependence, it means you have a defect in adhesion, which we can model simply by adding massless springs between the cell and the metal. We can calculate the theoretical acoustic reflection coefficient here that depends on the acoustic impedance of the metal, which we know, the acoustic impedance of the cell, and we also have a frequency dependent term here that comes from the presence of this massless spring. And we just fit our data with this model with two parameters, the acoustic impedance of the cell and the massless spring stiffness. And we're able to plot maps of the acoustic impedance and of the spring stiffness, which describes the quality of the interface. And in the case of the edge of the cell here, we see that we have a smaller, the interfacial stiffness is smaller near the nucleus, then increases in this intermediate region, and decreases again. Because actually the cell is not laying flat on the surface. The nucleus is like suspended by the fibers of the nucleus, and it's like floating above the surface, and then the fibers come down to the surface and pressing tightly to ensure mechanical stability to the nucleus, and that's why we have a better quality of adhesion of the cell in this region and a lower in this region. And on the edge, the cell is sensing the mechanical properties of the environment, so the very tip of the fibers are moving and very pressing softly on the surface just to probe the mechanical property of the environment. That's why we have a lower quality of adhesion at the edge. Skip that. And okay, as the last example of what we can do, we didn't talk too much about the ability of this setup to probe the thermal fluctuations, but this is also something we can do because we are heating the sample. So if we're able to record the temperature inside the metal film, we might be able to sense how much heat is absorbed by the cell, which is on the other side. So because of the heating, we have a bulging of the metallic film, which is of course exaggerated here, but if we send the probe beam on this surface, if there is no bulging, it will be reflected normally. You just send it back to a photodiode and you place an iris in between. If the beam is not defocused, it goes through the iris and you have a maximum voltage in the photodiode. If you have a bump, the probe beam will be slightly defocused and will be cut by the iris, and so the intensity on the photodiode will decrease. So you can measure the amplitude of this bump, which is directly proportional to the temperature inside the metallic film. So if there is a cell on the other side, part of the heat will escape to the cell and the temperature inside the metallic film will decrease. If I probe another area where there is no cell, the heat will be trapped inside the metallic film and the temperature inside the metal will increase. And so I just plot the amplitude and the phase.
So variation of temperature, what is, it is a few degrees?
Yes.
Few degrees?
Fraction of degree. And that's the way to control it. If I look at the amplitude and the frequency of this temperature change, as a function of frequency, if I'm at very low frequencies, I'll barely see anything. Because the temperature diffusing on such a large length that it goes through the film, but also through the cell and eats up the whole medium. If I go to sufficiently high frequencies, the heat is confined near the interface and we only see the contour of the cell. So if I want to avoid heating the cell too much, I can also increase the frequency and limit the penetration of the heat wave. Okay, so I will stop here. Thank you.
Thank you. Are there any additional questions? We're running a bit out of time.
Yes, just one question. How fast are you able to lock the laser?
How fast am I able to?
Are you able to lock the laser to stabilize the frequency of the laser?
The locking of the laser?
Yes, to keep the wavelength stable. Because I was looking at the cube of the figure of come back.
Back in which direction?
The last slide.
Ah, the last one?
Yes. On the top right side, the blue cube is output of the photodetector, right?
Yes.
And the red one is the derivative of that one.
No. The red one is the, so we are modulating the heat with an acoustic modulator. So we are modulating the heat at this frequency. And we are probing the defocusing of the probe beam with a locking amplifier at the same frequency. And the red curve is the voltage on the locking amplifier. This is to illustrate that when to maximize this effect, you need to defocus a little bit the beam initially, because if it's too well focused, the surface will move, but it will stay in the Rayleigh range of the beam, so you won't see anything. So you defocus it a little bit, and that's on the blue curve you see the photodiode signal. As you defocus it, when it's focused, the voltage on the photodiode is maximum, and when you defocus it, it goes to 0. But the lock-in signal is 0 when you're perfectly focused, because as I said, the movement of the surface remains in the Rayleigh range, you don't see anything. And when you start to defocus the beam, the voltage on the lock-in amplifier increases, and the maximum on the voltage of the lock-in corresponds to where the stop.
So the goal is to stay with the minimum of the lock. Let's say the lock-in should stay to 0, right?
No, it should be at maximum. You want the highest voltage as possible, to be as sensitive as possible. Because when you're here on the edge, so maximum voltage, any fluctuation in the position of this interface will be converted in change in voltage on the photodiode, because it will be cut by the iris. If you're there, well, it's completely focused, there's nothing to see. If you're at this position, all the beam goes through the iris, whatever the position. So you want to stay there to have the maximum voltage on the lock-in amplifier.
Good topic for the lunch. After, oh, sorry, one last question.
Okay, so thank you for a very fascinating talk on a very interesting topic. So coming from also optics and photonics, you can actually do optical traps, where basically you can put a cell and you can have a stiffness of your trap, and you can manipulate the cell, and you can also measure, let's say, a variety of mechanical properties of the cell, not, let's say, I would then say more freely suspended, because then it is not attached to anything. My question really is, I can see complementarities between those methods, but in your opinion, what is, if you would compare what you do to, let's say, the optical trapping and manipulation and that area, what is the complementarity that you obtain by this method?
I would say it's the frequency at which you can apply the deformation. With an optical trap, I guess you will just squeeze it slowly, quasi-statically, which is equivalent to the configuration when you have the cell in between two plates. Although with a trap, it's free. You don't have the plate, so it's free. But you cannot apply fast deformation. So you're only sensing the liquid parts of the rheological behavior of the cell, the low frequency part. If you want to sense the elastic regime, you need to go to higher frequencies. You need to apply a strength a bit rapidly, which is difficult, I think, to do with maybe it is possible, if you modulate the trap fast enough. I think that's the main advantage of this.
But the disadvantage is that you have some contact, so there's pros and cons perhaps, but I agree that the temporal regimes are different. So.
Yeah.
Maybe in the next step you could excite everything which is colored in the cell, like in optoacoustic imaging, and then look how the waves propagate, but I think it will destroy the cell.
I think some people are onto it. That's the idea. If you're able to design a transducer that is small enough, potentially the transducer that you can put inside the cell. But then, yeah, you need to take care of the heat of the plasmon excitation if you're using gold.
But people do that. I mean, you actually insert those nanoparticles into cells to actually.
Yeah, but with smaller with longer pulses. With femtosecond pulses, you will generate plasmon at the surface of the gold, and I'm not sure this is cell friendly.
Thank you once more.